In this presentation, I am going to present a demo on gene ontology using ShinyGo. So, the question is that what is Shiny? So, ShinyGo is basically a web-based tool used for gene ontology and functional analysis. Some unique features of ShinyGo includes that it displays the query genes on pathway diagrams and protein-protein interactions based on application programs interface access to KEG and string. We all know how popular is KEG and string for their pathway diagrams and protein-protein interactions. So ShinyGo has the access to those platforms, and it can show the pathway diagrams and protein-protein interactions by accessing to that those platforms also. Additionally, it can visualize the overlapping of the pathways, like there are genes that perform various genes perform various pathways, but how they are interacted, this can be visualized by the tool ShinyGo. It have another unique features, that is, it identifies the differences in gene type, length, GC content, and chromosomal distribution between queries genes and the background. So um, in the background we take all the other genes, like for example, we have taken the list of genes. So in ShinyGo we can visualize in which particular position is the gene present in the chromosome, or what is the gene type in respect of the other genes present in that chromosome, what is the contribution of that gene, how is the expression in comparison to the other genes present in the chromosome. So the tool ShinyGo was first published in the year 2020 by the university called Shell University in the journal Oxford Academic. It has 236 citations so far, and the paper says that ShinyGo is a graphical gene set enrichment tool for animals and plants. So, before proceeding to the tool demo, we should know what gene ontology means. So, gene ontology is a major bioinformatics initiative to unify the representation of gene and gene product attributes across all species. Gene ontology deals with, basically focuses on connecting the gene functionalities, gene functions and expressions across all species. A particular gene may be present in many species, but their contribution to the organism, their expression and all may differ. So, by gene ontology, it focuses on the connection in connecting the pathways of a particular gene in different species. Gene ontology is divided into three categories, namely molecular function, biological processes, and cellular components. Molecular functions categorizes the genes in respect of their molecular functions, like carbohydrate binding or ATPase activity. Biological processes categorizes the genes in according to their biological goals, such as mitosis or purine metabolism, that are accomplished by ordered assemblies of molecular functions. And the last is the cellular component. Cellular component is where the gene is present, the subcellular structure, location of the gene. For example, if it is present in nucleus or telomere, etc. So the topic I have chosen for the demo is autism. Autism is a neurological disbalance disease which is characterized by the difficulties in social interaction and communication and by restricted and repetitive behavior. So it is autism is a disease which is a combination of genetic and environmental factors. I have attached the paper of autism from oming.com. So it says it is inherited in isolated cases and it's multifactorial. It is neurologic. It affects the central nervous system and it is a behavioral psychiatric manifestation like it have difficulty in social interaction and have non-verbal behavior such as eye-to-eye gaze, facial expression, body posture and gestures. Um, so I want to study about this disease and see how the gene is interacting in this particular disease. What is the expression of the gene? What is the pathway? So let's proceed. I have prepared a flowchart of the demo. So at first we are going to search the term high functional autism in DisGNet. DisGNet is a database that have the collection of diseases, particularly genetic diseases. So I have chosen high functioning autism. Autism has a broad spectrum which includes low functioning and the mediocre functioning and high functioning. High functioning autism means those patients who knows how to read and write and basic social skills, who, who doesn't need any assistance or much assistance. So I will search about this term and then we would retrieve the data in Excel format. From the Excel format, we are going to copy the gene symbols and we are going to paste the data in the tool that is ShinyGo. And then we are going to analyze the data about how the genes are interacting in the pathways and what is their molecular function, biological processes, and cellular components. For the demo, I'm going to retrieve the data from this GNet. So I'm going to type this GNet here and enter. So we have got this website here. We are going to enter this and then go to search. And we have this column called diseases. Here I'm going to write autism and we have so many options of autism from where I'm going to select high functioning autism and search. So from here I'm going to go to the summary of disease and gene association. Here you can see how the disease is associated with the gene. Here we have 15 results. I'm going to download it. If you did not have ID here, you have to first log in to the website and then you can download it in Excel format. So it is being downloaded. It's downloaded. So we'll open the file and you can see that we have the table. I'm going to format it so that it looks clearer. So you can see we have the disease ID, genes associated, and what the gene does. Like this is the gene for oxytocin receptor, and this is the gene for binding protein 2, and so on. We have so many columns, like the protein class. From here now we are going to select the gene section and copy it. So we have copied the gene. So in another tab we are going to search for ShinyGo. We have the website here. We are going to click on the link and here we have a section called gene, just paste a list of genes and click submit. Here it says the ShinyGo is a gene ontology enrichment analysis plus more. So here we have sections like enrichment data, tree, network, gene, groups, plots, genome, promoters, string, and it is for the help section. And here we are just going to paste the gene. We have all the genes here, and we are going to submit it. So it will take some time and done, and you can see that we have the list of genes, and here from here we can select the human as we want only human, or we can select any organism we want, and then here it is the category what in which we want to display the gene functions. Like we have biological processes. For biological processes, the gene shows how uh, it is performing biological functions. Like this gene has a function of positive regulation of secretion. This one is for regulation of secretion by cell, positive regulation again, and then system processes cell to cell signaling, synapse assembly, and so on. And there are other options too. Here you can go to cellular components where you can see where it is present. That this gene is present in the plasma membrane region. The This gene is present in neuron projection. Then this in ion channel complex, cell projection, and so on. Additionally, we have molecular function to see how it is contributing to molecular level, like what the task it performs. Like this gene have the function of NAD plus nuclease activity, and this have NADP plus nucleosides activity, and so on. And we have additionally CAG section where we can view the cycle if there's, and in this data we don't have any, so we cannot show that, but otherwise you can see the pathway here, and there are many more. So here I'm going to select all available gene set, which would be a combination of everything. um, like we have positive regulation of secretion, which is under biological processes. Then we have signaling, social behavior, and etc. We have all mixed. Okay, so other section contains the tree. In tree, it shows how the functions are related to each other, like the regulation of secretion and the regulation of secretion by cell. The gene associated with these two are um, closely related. Then these three genes associated with its function, positive regulation of secretion of cell, positive regulation of secretion, and signal release. These are again closely related, and these two gene functions are closely related, and then it goes on. Like in the next section, we can see insulin secretion, regulation of insulin secretion, regulation of peptide hormone secretion, regulation of hormone secretion, regulation of protein secretion are closely related, which as a whole is closely related to this section over here, and so on. You can explore over here and know how it is related to each other. So this same table we can see with the network view. We can see how it is associated in terms of networks. You can see how it is associated, like synapse part is associated with this part, and then insulin regulation part, and if you want a clearer view of it, you can increase the age cutoff, and you can see how it is associated. Like see, this portion is fully associated, so closely associated that it is associated with cell-to-cell signaling than other sections. So we are going to keep it in 4 or 3. And then we can also change the layout however we are comfortable in. We can show it. See this layout, then this multiple layouts present here. See the association. Click on which will highlight. We will have the clearer view, highlighted view of the strings connected. So for download, we can go to static plot. Then we have download options. You can download it from here. We have wrap text where the text are wrapped in smaller section that takes small amount of space. Okay. Then we go to the gene section where we have the list of genes that we have uploaded along with the ensemble ID, symbol, gene type. See, we have just uploaded the genes, only the genes, and here we can get that this table which have and everything associated with the gene like the ensemble ID, symbols. The symbol we have already uploaded. So the ensemble ID, gene type. This is the all are protein genes. The species, then the chromosome number it has, in which it is present. Then position in the chromosome where it is present. So again, it has you can view the groups like for system process. We have these genes grouped together for regulation of biological quality. We have these genes grouped together and so on. We have a long list of genes here for the functions associated. Then we have plots which shows shows the expression of genes in the chromosomes. The distribution of genes actually. Let's see here. Number of axons, we have number of axons. Least time expected. How it is changing. Coding sequence length. This is the list one. And this is the genome expected. Transcript length, genome span, 5' UTR length, then 3' UTR length, GC content. You can all see how it differs from the expected range. And we have genome where you can see the position of the gene present in the chromosome. This gene over here is present in this position in chromosome 1. Then second we have this gene. Second we don't have any gene, and in third gene we have this particular gene over here in this position. So on, we have so many genes and their position in the chromosome. Here all the chromosomes are all the 22 chromosomes are present along with X chromosome. Then we have promoters, which we don't. Here we don't have any promoter site, so we cannot see. Here we have string. So here we are facing some problem. Maybe it's associated with the website. Otherwise, you can see the string data here. And over here we have the demo. Here it is written how it is curated the tool with examples like in human pathway database where the sources have been taken, where the data has been taken to make the particular tool. Let's see, there are so many things. We have a short demo to help section here. So that's it. Thank you, and I hope I could present the tool along with the demo. Thank you.